In terms of managing disease in the midterm, the obvious strategy here is breeding. We want to ultimately be able to deploy resistant lines in those areas that we know are going to get disease. We don't want to be depending on the chemicals that we can use in the short term, but ultimately we want the breeding to be able to deliver the solutions that we want to manage this disease. Again, we all know that there's increasing social licensing issues around using various chemical sprays, and we've got those options for the moment, but really we want to not have those be the only tool in our toolboxes. And the ultimate goal here is that we want to reduce losses, both economic. Obviously, if we can control the disease, we can minimise impacts on growth rates. But if we can minimise the dependence on chemical sprays, that's yet another expense that we don't need to worry about in managing our stands. And then in terms of the work that's been done around Coady dieback, that's ecological losses now that we're talking about as well. Some of the work that's been done in the Healthy Trees, Healthy Future program around screening for resistance to Coady dieback has involved screening seedlings at about age 15 months, and these get placed in these little buckets with inoculum in them, and they get re-exposed once a month. And then this is work done down at Manaki Whenua. They then wait and see which of them develop these characteristic lesions, suggesting that they've been infected. To date, I think the sense has generally been that there's not a lot of resistance that's been found to Coady dieback, but the outcomes from this study have been quite promising. When we look at 40 days post-inoculation, the red bars here, we're starting to see some families dropping off in terms of their survival rates, but it's at this 106 days post-inoculation. Some of these families here are almost completely wiped out. But the exciting thing is that there are some families at this other end of the spectrum that are not completely wiped out. And the fact that this is happening on a family basis, again, is suggesting that there could well be a genetic basis for this tolerance to Coady dieback as well. So this is a really exciting and really promising result. This has then been repeated with collections that have been done in subsequent years. Similar results were seen from the seedlings collected in 2017. The screening for the 2018 collections are still currently underway. This time the strategy was more to actively target within infected stands, to go and target the individuals that seem to be doing much better than the surrounding trees. And from that study, we've also done some preliminary genotyping work as well, trying to see if we can start to uncover some markers that might have correlations with resistance to this disease. The 2019 effort has just recently been completed as well in terms of doing collections. This was done in partnership with 15 different mana whenua groups. And much more, every year we've taken the learnings from the previous years, much more structured collections this time, both across and within the various rohe. And this is enabling a much more hierarchical analysis to be done of the results that we're going to hopefully get from these samples as well. This latest collection was an absolute mammoth effort. I can see Gordon sitting there in the audience. He'll tell you about all the cones that he received, and he had to manage processing. It's about 3.5 tonnes of cones that were received at Scion this year, and those then all had to have their seeds extracted and sorted, and yeah, this was an absolute massive effort. But this is going to provide a fantastic genetic resource that will enable long-term screening, selection and propagation to be done for this species. So this is absolutely hugely valuable. And again, collecting seeds from these trees is no trivial undertaking as well, with some of them, having to have climbers climb quite some serious heights up these trees to collect material. Jumping species to the horticultural side, so having a look at Phytophthora cactorium in terms of breeding options. The currently most widely used rootstock for apple, because most of the material is grafted onto rootstocks, is this individual over here. What was done at Plant and Food is that they then crossed this individual with another individual. This other blue bar here is the other parent, which has been used in a lot of their breeding to bring resistance to another disease, blight, and I think there's a few other diseases there as well. And then they screened using twig infections to see what the progeny of these two rootstock parents would look like. And again, lots of individuals in this batch or this side of the chart over here developing lots of lesions. But again, some really interesting findings down here where there are some individuals developing almost no lesions at all. And this is work that has been actively looked at Plant and Food. They've tentatively found some genetic markers that could associate with the resistance pattern as well. And this is a really exciting finding for the apple industry as well. But we're all here to know about pines, radiata pine and red needlecast. So I've presented at some of these meetings before and have said that we have found a genetic basis for red needlecast resistance in radiata pine. This has been done through a variety of assays, from field-based analyses to the artificial detached needle assay that was developed at Scion, as well as looking at shoot inoculations. We've developed some custom-built fogging chambers at Scion as well for doing inoculations, as well as just physically taking plants in pots and putting them out in infected stands and seeing what happens. And all of this work has shown that breeding is possible. Just the image down here, this was work that was done in the Radiata Pine Breeding Company's elite clone lines, where we screened around 400 individuals. This is lesion length over here on the y-axis. Most of them are sitting here in the middle. Most of them are average. It's what you expect. But it's the ones that are at the extremes, which are the ones that are interesting. Obviously, if you're breeding for resistance, you want to avoid the ones that are developing much longer lesion lengths on average. And these are the guys down here that would be of interest to the breeding program. And we've done some preliminary estimated breeding values based on the screening work, and those rankings have been provided to the RPBC. My understanding is that the material is still a bit too young to have pollen to do crossings with, but this is certainly information that the breeding program has now got in its pocket with which to make decisions. In the background, we're doing ongoing validations of all this work with field trial data. There's no substitute ultimately for a good solid outbreak in a natural stand. We've done the best we can with all these various artificial systems, and hopefully we'll eventually be able to completely close the circle with all these various tools and have some really solid knowledge around true resistance in our breeding populations. In terms of, Lindsay alluded to the fact that physiological needle blight is also caused by Phytophthora disease, and on a recce out to one of the elite clones field trials in Kinleith last year, myself and Nari Williams noticed some characteristic lesions on some of the trees that were out there, and these looked to be like physiological or Phytophthora needle blight. This was then given a full visual assessment in December 2018 and showed to have quite high correlation with Dothostroma needle blight, which had been assessed in previous years. Probably not too unsurprising, that result, because the symptoms of those two, they're both blights and their symptoms are fairly similar. But again, really high heritability for PNB resistance in this trial as well. So if we're going to start making selections for Dothostroma resistance, chances are that we're going to simultaneously and inadvertently, because of the confounding of the symptoms, be making gains for physiological Phytophthora needle blight as well. I mean, you can just see down here in the trial, really sick, blighted trees growing immediately adjacent to a very healthy green tree. So again, really strongly supporting the fact that we have got a genetic basis for resistance to this disease. So just in conclusion, we've got variability. That's, step one if you want to breed for anything, is you've got to have natural variability from which you can pick your extremes to make gains in the next generation. And we've got this both in radiata pine as well as kauri and the apple rootstocks as well. This means that we have got potential to breed, and that in the midterm we hope to have that there will be some strategies to help get resistant material to these various Phytophthora diseases. And in terms of the radiata pine picture, I said we've just demonstrated that we are having individuals with extremes of resistance or tolerance to red needlecast. These breeding values have been provided to the RPBC, and the really cool kind of unintended finding as well is that there is quite good correlation between PNB and Dothy resistance as well. The next step is to start to get this material out to the people in this room a lot quicker, and hopefully there are some tools in our tissue culture partnership work, which my colleague Jana will talk to you a bit, I think it's after the tea break, some other cool, exciting tools and technologies, just so that we can get this germplasm out to the industry faster. And on that, I will hand over to Rebecca. Thank you.